so much for joining us today. Welcome to our latest webinar series, exploring nanomaterial synthesis using liquid phase transmission electron microscopy. My name is Jen McConnell, and I'm the product manager here at Protochips, and we are excited to have such an elite and experienced lineup of speakers for this series. This series is going to explore different facets of synthesizing and characterizing new materials using our Poseidon AX system, which can also be directly translated to our new Triton AX system. Both are liquid cells for the TEM, but the new Triton AX system has been released with a uniquely wide temperature range, allowing for minus 50 degrees C up to 300 degrees C to be used during experiments in situ. So now, studying materials that form at higher boiling point or in higher boiling point solvents is possible, as well as exploring how low temperatures can protect more sensitive materials from the beam. You can read more about the Triton AX system in the brochure that's linked in the materials, or it may be called handouts, in the GoToWebinar platform. So again, there are several experienced speakers in this series using liquid phase TEM for these types of studies for many, many years. So be sure to sign up for all the others so you get reminded when they're coming up, and there's a link in the chat for those. So a couple housekeeping items before I introduce our first speaker, keynote speaker of the series. Everyone's going to be muted due to audience size. So please enter questions in the questions box in the GoToWebinar panel. At the end, we'll do a Q&A session with our speaker, and I'll read the questions from there. We also will have a survey that will pop up at the end. It'll only take a moment to fill out. If you wouldn't mind, it would be greatly appreciated and very helpful for us. Okay, let's get started. Let me introduce today's webinar presenter. We are absolutely honored to have you, Professor Gianneschi, to kick off this webinar series today surrounding the value of liquid phase TEM for hydrogel structure elucidation and property prediction. Let me tell you a little bit about Professor Nathan Gianneschi. He received his Bachelor of Science at the University of Adelaide in Australia in 1999 under Louis Rendina. In 2005, he completed his PhD at Northwestern University with Chad Merkin. Following a Dow Chemical postdoctoral fellowship at the Scripps Research Institute with Ray Zagadiri, in 2008, he began his independent career at the University of California, San Diego, where until June 2017, he was Teddy Traylor Scholar and Professor of Chemistry and Biochemistry, Nanoengineering, and Materials Science and Engineering. In July of 2017, Professor Gianneschi moved his research group to Northwestern University, where he is currently a Jacob and Rosalind Cohn Professor of Chemistry, Materials Science and Engineering, Biomedical Engineering, and Pharmacology. Professor Gianneschi takes an interdisciplinary approach to nanomaterials research with a focus on multifunctional materials with interests that include biomedical applications, programmed interactions with biomolecules in cells, and basic research into nanoscale materials design, synthesis, and characterization, including via new electron microscopy methods. So key areas of focus include the development of mimetic biological materials through chemical and nanoscale synthesis, including the development of protein mimetics and mimetics of melanin pigments. Professor Gianneschi is co-founder of multiple startup companies pursuing clinical translation of these technologies, including for the treatment of cancer. For his work, he has been awarded the Kabler Young Investigator Award for Nanoscience and Nanomedicine, the NIH Director's New Innovator Award, the NIH Director's Transformative Research Award, and last but certainly not least, the White House's highest honor for young scientists and engineers with the Presidential Early Career Award for Scientists and Engineers. And congratulations to you, Nathan, on that. Professor Gianneschi was awarded a Dreyfus Foundation Fellowship, is a Kavli Fellow of the National Academy of Sciences, a Fellow of the Royal Society of Chemistry, and is an Alfred P. Sloan Foundation Fellow. So again, Nathan, thank you so much for joining us. We really are excited to have you and your expertise here today and kicking this off for us. Please, by all means, whenever you're ready, let's go ahead and get started, and I'll come back for the Q&A at the end.
Okay, and you can hear me okay?
Yes, perfectly.
Okay, excellent. We'll get started. Thank you very much for that overly generous introduction, and it's really nice to be here with you guys. And this field of research for us has been a real passion over the last decade or so. Our particular interest is, as you can tell from the title, is in studying soft materials using these amazing tools. So both, of course, the electron microscope itself, but also these liquid holders that have kind of evolved over time to allow us to do all kinds of interesting things. Primarily, as I'll describe today, our interest has been in studying transformations, so chemical in some cases. In the case I'll talk about today, physical transformations. One of our long-term goals when we kicked this off, about a decade ago, was to study hydrogel structure. This presents a challenge if we look at, say, a dry state, dried out gel architecture for a number of different reasons. The predominant one, and this is general to other kinds of structures like this as well, is that this architecture itself is dependent on its solvated state. And that would mean necessarily that you must have changed it during processing, breaking up the gel, for example, to examine it by even cryo transmission electron microscopy methods. And so it stood out to us both in terms of a target structure that would be interesting to look at, what might we learn about the architectures, and then way beyond that, how could we study, for example, particle interactions with gel matrices in real time. And so you can imagine different potential applications, including the basic science of architecture and then, for example, diffusion through such matrices. So those are the types of questions we were asking. I'm gonna come back to that in a second and start off with perhaps our other, if I can click on this, that moves. Yeah, perhaps our other dream here in terms of really being able to develop, let's say, movies, for example, movies with electrons, as the slide says. You know, I've used this slide on and off over the years, but I think it kind of captures something of what we're thinking of, at least in our group, and I think amongst others in the field as well, is this fascination with going from static images that have lots of information, especially detailed information in real space, and also, of course, through other kinds of methods, chemical selectivity and spectroscopy included, that the movies themselves, right? There's a lot of information that's lost if you look purely at static images. Of course, there are many, many challenges with doing this with electrons that aren't met with the, let's say, the analog here, which is obviously from photography, standard photography. The left-hand side, for those interested, is apparently the first movie ever made. This was taken with a whole lot of different cameras lined up. You can see the numbers ticking behind the bottom left. A whole lot of cameras lined up and then triggered at a race course in California. And you can see that at the end of the 19th century. This described the mechanism, if you like, of horses at running at stride. On the right-hand side is another example, not of a movie, but of a flash photograph of a dynamic process. Of course, in this mode, under flash photography, stroboscopic type photography, the exact arrival time and then exit time of that bullet is known. And so with some technology around cameras and shutter speeds, etc., you could take images like this and many others like it. So what we'd love to do, I think, is something between both of these, or maybe both of these at the same time, but for unknown chemical systems at the nanoscale, which presents a lot of challenges. The main one being that we're damaging these samples as we're imaging them. And so we need to both pick interesting experiments to do and develop methods for mitigating things like damage, or at least understanding the role of the beam in these processes that we aim to study dynamically. As I said, the topic for today will be our efforts directed towards, let's say, capturing a real space image of these kinds of network architectures, for which there are all kinds of different models that describe how, let's say, the bulk properties of a gel, in this particular case, a hydrogel, are determined by, or perhaps predicted if you were to know these different pieces of architecture. Let's say the persistence length, for example, entanglement lengths, mesh sizes, concentration of the polymer, of course. If I knew those things and I understood the structure, could I ultimately predict, let's say, performance or behavior of the material? And that would be not just for hydrogels, but any kind of material, the type of thing we would want to know from let's say, a direct imaging experiment, combined, of course, with indirect methods, and I'll describe that. We got into this thinking somewhat, I think, agnostically initially about the types of gels we would look at. My student and co-author on this is a manuscript that's out for review currently, but he looked at a whole lot of different candidates. One of the features of the liquid cell, and I'll show a movie in a second, of course, many of you know this already, is that we can load free-flowing liquid samples quite easily. And that meant these lower critical solution temperature hydrogel materials became excellent candidates. They also, this particular form, methyl cellulose, is also a commodity kind of material used in all kinds of food products, building materials, pharmaceuticals, and drug delivery. And so it has some broad utility and interest. It's sort of a classical kind of material, if you like. The LCST, of course, is manifest in this case as the desolvation of that backbone. In other words, entropically favorable desolvation at high temperature that leads to the gelation. So clearly the polymer is aggregating or assembling in some way in 3 dimensions to form the gel network. And you can see that in the sort of classic upside down vial there, where the gel has formed under heating for some period of time. And the rheology on the right-hand side speaks to the same kind of thermal properties of the material. Again, the expedience here being that we can load a liquid and then we can heat the cell up using our in this case, a protochips holder that lets us get up to at least somewhere close to 100 degrees, in this case around 65 degrees, in order to form the gel in situ and then study it. In the bulk, as I showed above on the prior slide, we can do this quite routinely, of course, in the lab, and we can prep classic cryo TEM samples, and you can see the micrographs there. A, completely lacking information, just like in C. In other words, these are blank grids as expected for a free-flowing solution of polymer. We see no contrast, we see no material. In B, we've prepped a sample for cryo TEM from the middle sample. So in other words, from the heated sample. And what we see is commonly found in the literature as well. These are fibrils, approximately, call it 20 nanometers in diameter. We see the same kind of thermal gelation process when we study these by X-ray scattering as well, and that's depicted on this slide. The questions we had for this, and as I'll describe to you, was simply, is this correct, right? Is the structure of that gel really these 20 nanometer fibrils or some other kind of architecture? The origin of that question really comes from the physical reality of making the sample, and that is that you've formed the gel in bulk, and then you've no doubt disrupted and sheared it in order to study it by the cryo method. Not, of course, that you've cooled it, this is an instantaneous cooling process, but rather that you've processed the sample in some way. It may be cooling and breaking up, but it's certainly being destroyed in order to form that image or to make the micrograph that's then imaged in B. And this would be true for other kinds of materials. And as I said at the top, we're interested in a whole array of systems for which there might be unique opportunities by liquid phase transmission electron microscopy. And if not unique, perhaps liquid phase TEM could be better suited, let's say, to capturing these kinds of properties of materials compared to other kinds of methods. One would be simply materials in motion, as I said, diffusing species. Another might be non-aqueous systems that don't yield to cryogenic preservation, for example, as does the vitrification of water. And finally, most relevant to this discussion is responsive materials, or materials that have structure depending on the solvated state and or the temperature. And so, of course, the gel candidate that I'm describing fits both of those categories. This is the same type of setup, but just shown in a slightly different way. So we have two options, and we've used both of these options of loading sample, both what Molly did and what we're showing in the cartoon video on the top. And we can also use the flow cell. So in other words, we can flow the liquid into the cell. So this could be a completely sealed system in this case, and then during imaging, or rather in the microscope, we can fill the cell, in this case with a free-flowing, say, hydrogel, and then heat it at will. We can also do multi-component reactions using that same approach, and of course, us and many others have done it that way. Now, a basic reality of any of these types of experiments, dry state, cryo, or in the liquid phase, is that we are trying to, of course, develop an image at a given dose. In our case, these are very, very low dose experiments in terms of electrons per angstrom squared per second, but regardless, we're dealing with damage. So we're trying to form an image, trying to study material dynamically, hopefully capturing things that would also exist in let's say, real life or outside of the liquid cell, that would then correlate, either correlate, I should say, with a bulk solution experiment, or wherein we can confirm that the chemical structure isn't perturbed over the time of imaging during the experiment. So that's the type of thing that we've been concerned with over the years. This comes from a review article we wrote several years ago when everybody else was writing reviews in 2020, where we took a careful look at all the literature, including some of our own experiments, but tried to describe how these methods may be useful in this case to a polymer chemistry community in this particular case. You don't have to squint at all of this. Suffice to say that we're damaging the windows, or we're changing the window chemistry in some way. We're charging it. We're, of course, damaging the solvent. So no matter what kind of sample you're looking at, in our case, as I said, we're very interested in soft materials that are susceptible to damage, but the liquid is too, right? Whether it's an organic solvent, whether it's water. And over the years, we and many others now have cared deeply about this and have tried to model it for different types of solvent mixtures, and indeed try to quantify and then obviously use those models to quantify the kinds of damage that might be expected in the presence of a solute or an inorganic particle or organic polymer, for example. And of course, we have to deal in our case with damage to the sample. And so we're expecting things like cross-linking, chain scission. So if we're looking at polymers or biomolecules, we're looking for methods like mass spectrometry post-mortem in order to verify the extent of damage, or say functional group changes in the functional groups on the materials that would tell us that we've altered the sample in some way that above a certain, let's say, cumulative dose or at an instantaneous dose of a given amount of electrons per angstrom squared per second, that the results may be reliable or not. And that's the type of thing that we're reflecting on all the time. Now, as I showed you, these modern commercially available liquid cells and holders obviously use technology that I'll come to in 1 second, but this is by no means a new curiosity, right? Not only have these commercial systems been around for some time now and really inspired a whole lot of work, and increasingly so, and I'll show you a diagram that kind of depicts the beginning of that, but really since the start of the field in terms of examining materials generally by electron microscopy, there's been a fascination with this concept of studying liquid specimens. Frances, I have to give a thank you to her. She made these sort of historical perspective slides at a Gordon research conference we hold on liquid phase TEM, which is, by the way, open now for abstracts and things for February as well. She first presented this, and it gives a little bit of a context. A lot of the deficiencies in these measurements really came from our ability to make thin enough windows, small enough windows in liquid cells in order to study materials in liquids with even a hope of getting enough contrast to observe the kinds of things I'll show you in a second. And of course, this was the prime example of this. This is early days in 2003 that Frances and her team at IBM originally published this, which was a glued together version of one of these liquid cells that now the follow-up designs are based on. And so I should mention others that look at these in terms of capturing thin films of water and graphene. Our work, and as I've put some names here, and I'm sorry if I've excluded some other people, but these are some of the folks around the world who've now used either one of these systems. Our entire focus and the focus of today will be on these silicon nitride-based liquid cell setups. The field has expanded since that date. So I've ignored the 1940s here and just said since the insight around this initial silicon nitride cell from Francis Ross, the field's expanded. On the top, I've included examples, kind of a little bit biased towards our own examples of, because we wrote the paper, on organic materials, and on the bottom, examples of inorganic nanoparticles, which by far dominate the field because the materials themselves are obviously less sensitive to the beam. What we've done, as I said, mostly with organic or in some cases organic metal, organic framework type materials, is study dynamics. And so this would be things like the architecture of the material, just actually without moving, without undergoing any kinds of changes. This is what we refer to as imaging mode number 1. So this would be akin to like a cryo-TEM experiment where you're just purely looking at morphology. In this case, unilamellar vesicles, for example, on the top right. The left image is actually of a commercially available soap that many of you would know. This was done under highly viscous conditions. You can see multilamellar vesicles. So these are kind of, let's say, the liquid phase analog of a cryo TEM experiment, we are purely looking for morphology. Imaging particles in motion is exceptionally difficult because of the confinement problems and adherence of particles to the surface. This example from the Mojave group in Denmark, of course, and many of you know this team. It's a particularly good example of studying particle dynamics, but you can still see that many of the particles are stuck to the surface. So there's some challenges in terms of, and this isn't just camera speed, but it's also the confinement of the cell in terms of free motion. This has been used in our third imaging mode in terms of looking at physical transformation. So this would be inducing, say, shear or heating a sample up and looking at coalescence. And we've published several examples of that. And the final imaging mode will be where we're looking at chemical transformations, many of which over the years have been electron, say, beam-induced chemical transformations. We've also done some work on, say, metal and ligand, flowing them together in the liquid cell and looking at transformations in real time to measure things like kinetics and mechanism of formation of nanotubes and particles. This is an example of a kind of a mode 1 imaging experiment. We're simply looking at, in this case, a really simple emulsion. This is a water in oil emulsion. You can see that the higher density water in this sort of beautiful hexagonal array, in this case of a span 80 cyclohexane, you can see the phosphate buffer conditions on the left-hand side. This is the kind of dispersion that we're interested in for doing in situ polymerization, so emulsion polymerization, sorry. So we're interested in studying those kinds of multiphasic systems. It's possible under these conditions. This is an example of looking at coalescence of those other types, I should say, of emulsions. So these obviously dynamically would be impossible by other methods. We can study energetics of these processes. We can study surfactant chemistry, or let's say the propensity of a given surfactant to break up these emulsions or to stabilize them, and we can look at different mechanisms. In this case of coalescence, we can capture that process that, you know, hundreds of frames per second, for example, with excellent contrast. So these are the kind of reverse emulsion, sort of the beginnings of looking at dynamics, just as an example of that. I've always liked this image as you're sort of capturing that moment of coalescence between these particles, and that can be looked into more deeply. We can look at things like the inverse of that. So this is a standard emulsion, so an oil and water, if you like. And in motion, of course, you can see these low contrast white sort of particles moving in the field. Even in this case, despite, and obviously, and interestingly, as soon as it stops moving, the human eye finds it very difficult to see that motion. I'm going to play that again so you can sort of see them moving, I hope, on your screen. And we've studied this extensively. This is now a paper that's out for revision and potential rejection as we speak. We just submitted it yesterday. But this was us trying to contend with anomalous motion in a liquid cell and try to build some models that other people can use to sort of understand where and when the motion of the particle itself is damaged by, let's say, not just the beam or influenced by the beam, but also influenced by being in the liquid cell. And you can't see it in this image, but we're imaging the top right corner. You can kind of see that in the shadow here on the top right. These particles tend to be moving actually over time towards the center of this cell where there's a bulk. And so we can see that kind of directionally biased motion in these experiments. This is an example of where we've used an in situ stain. So this is a nickel chloride stain that we use to actually stain these vesicles. On the right-hand side is the liquid cell image or movie that I showed you earlier, purely looking at, characterizing them in terms of being, as you can see, fairly nice unilamellar vesicles. Those same vesicles by dry state TEM are shown on the left, of course, kind of crenated and crushed down to form that. But these are identical materials, just imaged by the liquid cell or standard negative stain imaging with the urinal acetate, as you can see. I'll quickly show you this. This is a movie I showed on the other slide, but this was where we're able to capture dynamic motion. These are micelle fusion. So these are particle collisions resulting in phase transitions that we're able to observe. These were really fortuitous experiments that are difficult to repeat, but they allowed us to sort of model and understand some of the energetics of these kinds of fusion reactions. So these are just examples, in this case, a physical transformation caused by shear in the liquid cell, of the types of things we can examine by these methods. In this particular case, we have no idea the extent to which we're damaging that polymer that's shown in A. The extent to which the beam itself is inducing that process was just not understood at that time. Since then, we've endeavored to develop tools in parallel with the types of experiments I'll show you in a minute to really then go back post-mortem, so following the experiment itself, to verify whether or not we've damaged, in our case, the polymer. I should say, if you're looking at crystalline materials like the metal organic frameworks or other kinds of crystalline materials that we make, you can, of course, verify that you have or haven't damaged the structure post-mortem using electron diffraction. In this case, we can't. These are sort of, let's say, non-crystalline polymer materials, or peptides was a particular example of self-assembling peptides that we first published in this manuscript in 2019. The procedure is relatively straightforward if you have this moldy imaging mass spectrometer. So that's the catch. You've got to have that instrument. We load the sample. We image the sample, as you can see in the electron beam. We crack the cell open. There's probably some motion, but limited amount. It dries pretty rapidly. It's only, remember, a few 100 nanometer at most film of water. We matrix, so this is an automated sprayer of the moldy matrix, which assists, of course, in the ionization process. We use a laser source that has a footprint that's much smaller than the size of the window. So we can scan across the window and prove that polymer that we imaged is not damaged. And of course, we can use a reference, which is outside of the window, in order to determine that where damage has occurred. And we generate these damage plots where at a given cumulative dose and instantaneous dose, we can plot that and determine where we're in, let's call it safe imaging conditions, or where we deem it arbitrarily acceptable to image up until a given dose for a given amount of time. An example of that that was never published, it's just not particularly interesting, but is a nice demonstration for a talk, is this NIPAM polymer. So this is a polymer with an LCST like the hydrogels I'm about to tell you about, where we've simply loaded the cell. You're imaging at one of the corners here. We heat to 45 degrees and wait, and we see this desolvation event. So here you see basically chunks of this polymer that's essentially precipitated in the cell. We've desolvated the cell in this case. We can cool it back down, give it some time. We resolvate, maybe not 100% reversibly, but over the course of this, of course, we can change the temperature and the dose rates. And then we can, in this particular example at 0.1 electrons per angstrom squared per second, using this, obviously we weren't continuously imaging, we're just imaging occasionally to capture these different frames, and then measure by mass spectrometry at the end and prove that at least that there is some NIPAM present and that the molecular weight didn't change. And in addition that the dispersity didn't change. So the shape of this curve, in other words, the envelope of polymer didn't change. We've done some other things like this kind of correlative imaging, combining, of course, multiple methods. One would like to, for example, do things like light scattering, X-ray scattering in a sample, maybe in capillaries, neutron scattering under similar conditions, and certainly electron scattering type experiments of the type that I've just mentioned. This was an image that came from Cheng Wang's group at ALS, who we worked with on this, sorry, I skipped forward a slide, where he had proposed mounting these tips, and you can see that liquid cell tip mounted in his RSOX holder at Berkeley, where we could do an experiment, and we published on this in 2023, where we did an experiment. We shipped samples out, did experiments at Berkeley on the beamline, where we did these soft X-ray spectroscopy measurements at different temperatures in exactly the same sample geometry as we could do in the liquid cell holder, not at the lab at Berkeley, but at Northwestern. So it's not exactly a correlative experiment. You'd love to take the holder and stick it in the microscope and go back and forth. But in lieu of that kind of multi-million-dollar experiment, this was a demonstration of our ability to examine phase transitions that were temperature induced, where we could pin it not just on, in other words, by two completely different methods, and then tease out where we had electron damage, et cetera, or rather where we were imaging within a safe regime by correlating the X-ray result with the electron microscopy result. Okay, so this is a bit of background as to how we think about using correlative imaging measurements to verify whether or not we've damaged a sample, and then either throw out a complete idea, right? If the samples are just too sensitive, we may not continue to study them, or that will allow us to pick an imaging window of time and dose that we consider safe. Okay, so that's the kind of core of how these experiments are done, at least for these kinds of materials. Okay, let's go back to the slide I showed you earlier. So this is the same slide I said earlier. This was a bulk solution experiment, heating up to make a gel, cooling down to disperse the polymer, and then capturing and shearing this gel at that heated point and getting a cryo-TEM image. This is exactly replicating what had been shown in literature. And just for, if you want to look on the top right, that's the memory, that's to remind you of the structure of the methyl, say, modified cellulosic type material that we're looking at. Okay, by liquid cell TEM, so my student Nathan Rosenmann went away and did a whole lot of experiments, and this was probably one of the most exciting set of micrographs I've seen in the decade or so of doing this work. What he's done here is, again, he's imaging at this corner of this window. You can see it completely, you see nothing initially. So this was at a dose, a cumulative dose of 0.2 electrons per angstrom squared. He's then blanked the beam. He's heated it to 65 degrees and then imaged again in that same corner. So he's added another 0.2 electrons per angstrom squared. You can see the dose rate to the bottom here that's consistently used. He's then blanked the beam, allowed it to cool down, and you can see sort of the timestamp here over this period of time. Now at 80 minutes in the full experiment from the initial time, he's then seeing this annealing. And you saw that in the hysteresis, if you remember from that data, where it takes a long time to redisperse. And indeed, in some cases, we need to reduce it below 25 degrees to redisperse these polymers. But you see this really beautiful micrograph in the middle that I want you to focus on, which show these fibrils, what look like individual fibrils, so bundles of polymer, then bundling to make these bundles of fibril, which we would call fibers. So let's do it again. So go back in the lab and do this again was sort of the consensus. This is the right-hand image. This is the same one that I showed you earlier, but he took that same procedure and repeated it multiple times. And we see this same kind of effect. So this is showing you the sort of fiber diameter, if you like, made up of these individual 20 nanometer fibrils. And you can see that here in this case as well. This sort of 1 to 2 micron, let's say, diameter fibers. This is a whole set of more micrographs here showing you at 65 degrees at these particular time points. This is multiple different experiments showing you these fibers as they're growing. I'll very quickly mention that he also used a micro FTIR measurement of these to look at maintenance of the functional groups along the polysaccharide. This was done instead of some mass spectrometry that I mentioned because those polymers didn't fly by those methods. So we're using these kinds of correlative approaches after the experiment to determine that we are or aren't in that safe imaging mode. This experiment that was then done, this is under continuous flux. So this is an instantaneous flux, I should say, of 0.17 electrons per angstrom squared per second. And on the top right, you'll see the cumulative dose, temperature on the top left, and the time. You can see he's at 25 degrees centigrade. He's going to the top right and then back down to the bottom right. He's now at 65 degrees. So you can see the time's gone by now. And you can see the beginning of this really beautiful set. By the way, this is just detritus on the outside of the cell. This is not the material that you see there, that little thing on the left-hand side. The material is coming into view as he's imaging it. You can see that cumulative dose. He's going to get up to something like 180 electrons per angstrom squared, where we say we're probably at the edge of where we can reliably or pseudo-reliably determine that we're not destroying the material. So we're safe until that point, we're saying. You can see here, and the image is flickering around, but we can kind of see now these bundles of fibers projecting from that center point. This is now a cleaned up set of micrographs for you to look at. You can see that, just ignore that debris on the left-hand side. So this is at 0.2 electrons per angstrom squared, as I showed you on the prior slide. We're breaking that movie out into these different panels. Here's at 65 degrees at 30 minutes, and you can see now that really beautiful set of bundled fibrils. And now instead of being a plate of boiled spaghetti, those fibrils look like they're sort of packed together into these bundles. We've then cooled it over time, and we're approaching where we're probably damaging the material a little bit, but it's provocative to think that we might actually be seeing now the unwinding of those fibrils as we break up and disperse the material. Because of confinement and the timing, that may not be the case, but it's kind of interesting to think we might be able to see this reversible process in a future case, especially where we could cool the liquid cell completely below room temperature, which we can't do currently with our system. Shown here in the zoom up is one of these. We've zoomed up on this fibril, and this fiber, sorry, to show you one of these 20 nanometer fibrils, which of course we observe by cryo-TEM. So this is now shown side by side. So the cryo is not wrong. The gel is made up of these individual 20 nanometer fibrils. It's just that it's not the morphology. And we, at least from the liquid cell experiment, this would imply that in fact these bundle together to make these higher order fibers. Now, if we look at persistence length, which if you remember in the model is one of the physical parameters of the gel that we might be able to use to predict the gel properties, we can measure that, of course. And here, let's call it 4 microns persistent length. From the cryo measurement, let's say it's 10 times less than that. And so therefore, that should have a big influence, right? If we use the persistence length in those equations together with the concentration of the polymer, we would predict very different gel properties depending on which structure is right. Now, what we said here, of course, was, as I've written down, as actually Nathan made these slides, this is a potential artifact of the experiment, of course, right? So is this true, or are you making some completely unusual material in the liquid cell that just doesn't exist in any other state? We did a whole lot of experiments, he did a whole lot of experiments, including some high resolution fluorescence microscopy to try to capture whether or not we have these, first of all, larger micron scale bundles, and whether we have these large persistence linked materials. This was an experiment he then settled on. This was done together in our bio-cryo facility at Nuance at Northwestern with Rainer Blair. They went back in time, which is kind of always many times the best way to do science, looked at the older literature. So this comes from a study from 1982, looking at essentially freeze drying tissue samples. So this is some of the original diagrams. The concept being that you could sublimate off the ice of a non-blotted sample on a grid in the TEM in order to create a sample that more closely reflects a structure without having to blot it. And so this is just a more fancy diagram of exactly the same thing, where again, we're using the transmission electron microscope purely to sublimate off that little bit of liquid, and then hopefully leave behind this preserved, more three-dimensional sample. So you can see maybe where we're going with this. This is then inserted in an SEM in order to image that gel network that's preserved on the bottom right in panel H. And so now we're looking at that in an SEM, and we're saying, well, okay, these look aligned. They're on the 300 nanometers, sometimes maybe micron length scale, and certainly the fibrils themselves don't look like this kind of dispersed, randomly organized boiled spaghetti arrangement, but rather this kind of beautifully kind of woven fiber looking material. And you can particularly see that at the higher weight percent methyl cellulose sample here that look like you're zoomed in on a woolen blanket. This can then be analyzed. This was done together with another graduate student of mine, Lauren Erie at Northwestern, to look at, let's say, many fibrils and fibers from both the liquid cell experiments and our SEM measurements in order to get a better idea of persistence length across with better statistics. Again, we're looking, call it 3.5, 4 microns. By the SEM measurement of the freeze-dried sample, call it 2.5 microns, something like that. The cryo-EM measurement in that hundreds of range. Of course, a standard small angle neutron scattering measurement that's sensitive to this size regime is going to predict something closer to 50 nanometers. Again, none of this is wrong. It's just what the probe and the sample prep itself is looking at. And so went back and did the rheology and said, okay, well, let's see, right? Determine G prime at different temperatures, at different concentrations, and then see if your structural prediction of what should occur. In other words, if I know the persistence and I know the concentration of the polymer, and you're right about the liquid cell experiment and the SEM measurement, then you should be able to predict the G prime. Is that true? And so this is the experimental setup. In black here are those experimental data now plotted in concentration versus G prime. So that's the real rheology there, and then fit to the red curve. The prediction from the SEM measurement shown here. The LCTEM experiment is shown in purple. So we're getting much closer, and that's really highlighted on the right-hand side here. obviously the neutron scattering measurement is not going to be accurate. It's not interrogating the right length scale, right? You're not in the ultra small angle region where you would see these micron scale structures at all. In the cryo measurement, of course, is essentially modeling this as if it were a much weaker gel. Now, this can all be fixed with fudge factors. You don't need to do this experiment. The simple relationship is the G prime is proportional, of course, to the concentration of polymer with some fudge factor. So for a given material, I could experimentally determine this. The point is, we can use these direct measurements then to maybe predict something more accurately about, say, an unknown material, of course, and in terms of these structure-function relationships. How generalizable is this? This has been a difficult set of experiments, but we've now looked at this for other polysaccharide gels, so HPMC and HPC. You can sort of see the structures really briefly there. These form fibrils. HPC, to a lesser extent, is a hierarchical architecture. We can study these by, again, another method, which is now going into the ultra small angle range. So now we can interrogate these bundles that you'll see highlighted in terms of the data on the top right. These 1.6 to call it 3.5 micron. These are diameters of the bundles, not persistence lengths. But certainly the USENS data for methylcellulose is consistent with what we see by the freeze-dried SEM measurement, as I mentioned. We can also correlate this with small angle light scattering as well, which was done with Mike Hoare's group at Case Western. So by now correlating these bulk, call it scattering approaches, ultra small angle neutron, we've got the X-ray data plus SEM data, all instigated by these really beautiful structures that we're able to observe by liquid cell TEM, correlating to at least suggest that small 20 nanobit of fibril is not actually the gel forming unit, which is to say that if we had all this data for every kinds of material, all of these types of materials, we might learn something deeper about how the gels are formed. Just to give you an idea of that, this is now the SEI, let's call it the freeze-dried SEM data on the bottom right for the HPMC and HPC samples, showing again these kind of aligned woven fabric kind of structures that we saw for methylcellulose as well. So in summary, my postdoc at the time has now moved on to Texas A&M. Yu Chen made this nice diagram on top, kind of suggesting, well, look, all of this connects together, right? We need cryo-TEM to correctly interrogate these structures at these length scales, although the hierarchical structure isn't observed by that method because of the way the sample is made. If I keep it in a liquid cell, I kind of see, yeah, bundles of fibrils. There's limited, right? This space here is clearly not present in the bulk. Rather, this would be a dense array of interconnected bundles of fibrils in reality. Obviously, under the conditions of liquid cell, we see this initial bundling together with this kind of blank space. But at least it tells us that these length scales should be found, let's say, in bulk scattering measurements. And the freeze-dried kind of approach has allowed us to get to sort of that view, maybe, of the reality in the bulk. So I'm going to show you one more little movie here because I think it's kind of nicely done. Again, this was done by my postdoc at the time, Yu Chen. You can watch that on the left-hand side. It's not going to give you too much more information, but happy to provide these to anybody who'd like to use them in the presentations. The reality of this, and I think the real summary for this, that you know, our message anyway, is sort of imagine your materials here. This is a really nice review article that's still highly relevant today, really describing the importance of using correlative methods. You can see that AFM, electron tomography, cryo-TEM, all of those approaches together with scattering methods in order to determine the structure of, in particular, complex soft materials. And we would say that I think our work and that of many other groups has really looked at, well, what if you also had this ability to look at dynamics, solvated structure, and the liquid phase. And I think we could have convinced Joe Patterson and Rachel at the time, if we had a time machine, that maybe liquid phase TEM could be included. I think there was a single example in that manuscript at that time, and Joe could correct me if I'm wrong. But I think we've now, his group and others as well, have expanded the scope of this to say that I think that that method belongs in this sort of kaleidoscope of approaches to determining structure. I haven't really shown you this, but I think you can see where maybe the philosophy is. And we did make some movies, but really the static stroboscopic imaging really told a bit more of a story about the mechanism. But I think this was mostly about that idea of keeping a solvated material should be imaged, let's say, in its solvated state. That at least concept that we hope to get at with these hydrogel structures. I've mentioned Nathan a few times. I'll thank him again. I put a little asterisk next to his name. He really drove this project, did all of that work, just a huge amount of experimentation. I included Joanna on here. Joanna Carpentier was a graduate student of mine up until 23, and really initiated that project with Nathan. Other people on those on our TEM projects are shown in purple. Thank you to many, many collaborators for new materials, great ideas of the kinds of questions that would be interesting to material scientists, polymer chemists. We have collaborators on image analysis and others on other parts of how we describe these experiments as well, and I've shown them on the bottom left. Funding agencies, that's an older photo of my group that kind of gives you an idea of where we are when the sun's shining in Evanston, and I'm happy to take questions. I think we have at least 10 minutes.
Wonderful. Thank you so much, Nathan. That was a really nice presentation, some beautiful images, and I know you guys take on a lot of challenging samples. I mean, soft materials in general are challenging in liquid to begin with. So again, thank you for the presentation and kind of going through that. If anybody has questions, please put them in the questions box now. I do have a couple to start with, and then we'll go through and answer over the next handful of minutes. So again, these materials are challenging. We've got a lot of people interested in studying soft materials. Is there like an order of operations that you could recommend for somebody who is picking up a new soft material? They want to analyze it with TEM, in situ TEM, but like where do they start? You probably don't start with TEM. I'm sure a lot of these other either bulk scale or ex situ analyses may be best first. Like, what's the order of operations you would recommend for somebody new to soft materials?
I mean, that's a good question. So I think, you know, we've made some we've published a couple of papers where we kind of describe a bit of a workflow, but it's probably maybe best depicted on this sort of slide. And hopefully this answers it. Obviously there's a longer story, and I'm happy to talk to anybody about any of this, but like you said, maybe you're starting with a self-assembled particle or a polymeric nanoparticle or something that's on a reasonable length scale, right? That could be imaged by the method. So there's some physical constraints, right? You need to be able to image through the liquid. By the way, any solvent, right? These instruments are now compatible with many, many different solvents. And so there's less of a challenge on that side. But you're probably starting with an idea, let's say, from scattering methods and maybe traditional electron microscopy methods of the size and shape of the material. Now, in the case of some of the more, let's say, crystalline materials, you then have a whole other set of methods because you know that you might know something more about the actual structure of it. For amorphous materials, that's why we've kind of harped on about these other kinds of approaches. So in this particular example, we used obviously this kind of fancy setup at ALS, but really if you're looking at dynamic processes, you want to understand them by combinations of scattering and mass spectrometry and everything else that I described. Then you're asking a defined question then about a material. Let's say that all of that's true. I have a polymer-based micelle or a liposome or a self-assembled peptide or something that I'm interested in. If, for example, it were to yield to this kind of thing, which you could check, and if you had that instrument, then you're golden, right? If I'm making a crystalline material, I know I can image it, crack the cell open, do electron diffraction. I could do standard TEM in the dry state and verify structure. If it's a polymer or a protein or a peptide or something like that, I could verify the safe conditions, as in what dose rate and what cumulative dose can I be at and not damage the sample. And then I'm asking a question, right? In many ways, the hardest part of it is the question you want to ask, which is, for example, what is the mechanism of the phase transition, right? It's a moth undergoing some change in phase. It's in our case, right, we've done those kinds of things, or polymers or polymeric material undergoing a phase transition that's measurable in the bulk. So you know from scattering or some other measurement that you're going from one phase to another. You might want to directly analyze that so you can study that mechanism. So you kind of need to know the time course of that. And is it relevant, right? If it's in microseconds, you're not going to capture it, right? If it's over the course of several minutes, you maybe have a chance. So we have a lot of these conversations with potential collaborators where they'll say, well, I've got this really cool system. We've made this self-assembled material. And those are the types of things we'll look for, right? Well, what's the size of it? If it's a 2-micron scale rigid particle, we can't put it in the cell, right? So there's cell dimensions, right? We know we want to be on those hundreds of nanometers, or it can be squished down. So the emulsion particles can be larger because they can squeeze in between the, let's say, the plates. So there's kind of a size of particle, then there's a beam sensitivity of the particle, and then you can start interrogating something like temperature dependent structure over time.
Very good. Yeah, we get these questions a lot, and I think people will dive right into in situ TEM with a new material, and they're like, oh, it's damaged. I don't think this will work. But I really think there's a lot of workup you can do ahead of time to try and make that successful, you know?
I would say, actually, I didn't say it, but like you would know intuitively, the cryo experiment is key, right? Mm. If you don't get, I mean, if you get no contrast at all, you're not going to see it in liquid, right? So there's some simple things like that. I mean, you know, that, for example, these materials, this HPC materials, you can even see in the image there, it's extremely difficult to detect these fibrils by cryo-TEM. You can imagine we were unsuccessful in liquid phase. And that's right there, right? So just that question of contrast, because sometimes it isn't beam sensitivity. People, if you're not used to these kinds of experiments, you are normally these people are pounding these things with electrons. Most of these experiments are done at fractions of electrons per angstrom squared per second. So sometimes the dose is problematic, but a lot of times it's intrinsic contrast.
Gotcha. So the cryo is a great first screening. How good is your contrast in cryo to kind of give you an idea of where you might land?
That may be the same. Yeah.
Yeah. Very good. I have a follow-up question, but let me ask from the audience first. So there is somebody asking about sharing your PowerPoint with the audience. How do you feel about that? Is that okay?
I could do that. And the manuscript on this is in really close to publication. So I have no problem with that. All of this data is in there, and I'll eliminate some videos and things so it's not 15 gigabytes, and happy to share it.
Great. If you don't mind sharing it with me once you've done that, and I can share it to the entire attendee list.
I can make a PDF for you. Yep.
Perfect. That'd be wonderful. Thank you. And then the next question, what do you think regarding environmental scanning electron microscopes for soft material imaging, especially with no sample preparation needed before imaging?
Yeah, and look, we've done none of that. But there's no reason. The main reason is you just can't get around to everything. But I think the least impact you can have on that sample, the better, right? And I think we're dealing with problems of confinement. You know, I would argue that we're getting better resolution, but you know, you're still dealing with resolution problems that are inherent to the thickness of the liquid cell in the sample. So the easy answer for me, without having a conversation about it, would be, yeah, you know, all data helps, right? And it, of course, could be used correlatively, but may actually be superior because of exactly the reason that you're asking. There's just less sample prep and confinement problems.
Yeah, very good. I had a quick follow-up question and then one more after that. And if anybody else has any more questions, please put them in the questions box now and I can ask before our time is up. But with the fibrillar hydrogels, fibrillar, okay, that one. What does the damage look like? Because you know you're avoiding damage. How do you know what the damage looks like for these different materials when you're trying to figure out kind of where that benchmark is for your dose?
Yeah, so the way those experiments are done that I didn't talk through, I'll go here just so you can kind of see. We don't think, I was hedging a bit. We don't believe that this is damage. In other words, if we we can heat the cell without imaging at all, right? And then just open up the beam and get these kinds of images that you see on the top right. If we were to sit here on this and crank it, you know, either go past, you know, let's say to thousands of electrons per angstrom squared, we just start seeing open fields, as in it just looks like the blank, like the top left.
Gotcha.
So as you destroy the polymer, obviously we assume that we're losing molecular weight. We end up just breaking the material up. So they look like empty cells.
Gotcha.
In the infrared experiments and say by a mass spectrometry measurement, you just see no detectable. The problem is you see nothing detectable. It's not like you start, in this particular case, we don't see clumps and things, although I'm sure many other people have seen that type of thing where there's like wisps of the material now floating around randomly. I think in some ways, the long distance order on that micrograph on the top right is a good sign that you probably haven't damaged something. It's hard to accidentally get something like that, and then obviously consistently see it reproducibly. But yeah, the damage manifest in this case is like a dispersed sample, as in invisible.
Gotcha. Okay. And then we've got one more kind of general question, because again, like I said, we get a lot of questions, about soft materials and analyzing soft materials, and people are interested in liquid cell for it, but are just not quite sure. And you talked about this a little bit at the beginning, and so maybe we can reiterate, but with chemistry and synthesis being such a broad category, do you mind kind of going over once more, like the types of samples and processes that you think present the best opportunity for liquid phase TEM to answer like key questions that other techniques can't?
Yeah, I sort of had a couple of slides on this, and this goes back to, so Joe Patterson was a postdoc with me, now he's at UC Irvine, many of you know. We thought in the early days about our time with this anyway, in sort of 2014, 15, 16 period, about exactly that question. This is a list of things we thought of, right? Things that, as I said, unique opportunities, but maybe where the liquid cell can capture things that are quite difficult by cryo-TEM in particular. And of course, dynamics, although there are ways around that, obviously, by doing sort of freeze frame imaging, if you like, sort of mixed systems. So that's why we've picked on emulsions. We've put crude oil in the liquid cell and studied how different surfactants break up the crude, the particles. You can kind of imagine doing lots of things like that you just would never yield. Obviously the dynamics of that, but just even the material itself, you can't make a cryo sample. And gels have kind of popped out, but these types of things, I said it a few times, but I think anything where the structure is dependent on the solvated state and where the structure is dependent on being at a fixed temperature, just because it's so difficult to reliably make a cryo sample at a fixed high temperature. So I think if you're interested in those types of systems, right? So dynamics, emulsions being sort of mixed phase, things that are non-aqueous, and things that have either dynamics or structure that are temperature or solvent dependent. Those are good candidates. The other area, things like, we've had a lot of fun over the years with things like metal organic frameworks, because you can mix two components, right? The solvothermal syntheses, so you can mix metals and ligands and heat it up. The product is crystalline. So, anything where the product is indisputable in terms of its structure, then lets you study it dynamically and then even define the kind of beam damage that you observe. So those are the types of materials, I think, sort of generally. And then we've always made this description of, that there are 4 imaging modes. Maybe there are more, and maybe I'm wrong, but this is the way we think of it, right? Just static solvated structure. Most people making materials like this care about that. And that's what people have done with cryo-TEM. Do I have a multilamellar, unilamellar vesicle, etc.? Particles moving around, why do I care? It's a difficult experiment, but I might care about, the velocity of a collision that leads to a phase transition and something like that. Physical transformations is number 3, and then 4th would be, like I said, making materials through chemical processes. Hopefully that aren't beam induced, so that you can roll that out, although there's lots of interesting studies in that area as well. So I think there's both kind of keeping in mind what your question is. Do I care about morphology? Do I care about dynamics? And then of the types of materials, am I studying something unique by this method? And then importantly, am I really studying the materials, or am I studying some damaged product? And that's where the sort of safe imaging mode comes in.
Very good. Thank you. I know you talked a bit about it in your talk, but it's just I think it was important for those that came in late, and for those that are going to rewatch later, to just emphasize. But
no, I think that's a good point. I should probably come back to the end. That's the most important thing, right? That this is a system we happen to care about, but I think it has got to the point where certainly mode 1, right, should be routine in most microscopy facilities around the country and overseas. And I think there's a lot more to be done in that kind of space alone.
Yeah, we definitely agree. Well, again, we have gotten to time here. Thank you so much, Nathan. I know you're a busy man and really appreciate you taking some time to present for us and for the community as well. We'll put this on YouTube so it can be rewatched and share, and it'll go in our success community as well. So again, thank you very much. And we'll send the PowerPoint whenever you have a chance to share it with us. We'll send it out to everybody. But
Will do. Yep,
yeah.
I'll try to compress it. I'll probably have to get Nathan to compress it because I don't know what I'm doing. But,
he's a smart guy. He'll figure it out.
I'll make like a 25 gigabyte PDF in a minute. But anyway, yeah, I'll do it.
Well, we'll look forward to it, and we will talk with you soon. Have a great evening, day, morning, everybody, and we'll see you at the next episode. Thanks, Nathan.
Thank you. See ya.
Bye.
How do I exit?
I will exit it for you.